Okay, Benji, so I'm going to try and share the screen out again.
Yeah.
Here we go. Okay, so this was the poster I created from the Hobson's Conduit eDNA survey of Hobson's Brook, and we were quickly looking at vertebrates and really useful. The 6 samples produced 11 species of fish. We thought we had 3 or 4, so that was really excellent. A couple of surprises in there, like the introduced eyed, which is a kind of little goldfish, and the pike, which we thought the stream was too small to support, but it's clearly in there. And there were some notable absentees, which we hoped we had, for example, eel and trout, brown trout. But that was fantastic. And we found smooth newt from the eDNA, which we didn't know we had. So that was really great. Not many surprises in the mammals. And in the birds, perhaps we were surprised that there were so few species of bird, but the real one that stood out for us was the water rail, which we weren't aware of. They're obviously very secretive birds. So from our perspective, this 6 sample eDNA survey using your methodology has worked really, really well. It's opened up all kinds of possibilities for us, including now we know about the fish, we'll be able to ask for funding to put in a fish pass, which will connect this chalk stream to the River Cam to allow some migratory fish to come up. So really, really fantastic. Some of the questions that I was asked during the BioBlitz event that we recently had involved why aren't you seeing human DNA, and why aren't you seeing dog and cat DNA? And I thought perhaps you'd be able to answer those.
Yeah, of course. Yeah, no, those are good questions. There should be probably a small note on the report, but basically we exclude all sort of contaminants, sort of common contaminant DNA. So human is considered, it's pervasive everywhere, even when we go to the most remote areas in the world to work. So we do just we remove the human DNA that we pick up. It's omnipresent. We will alert you if there is a very high prevalence of human DNA, because it can interfere with the results. But if it's sort of below our threshold, then we don't report it. There should be a note in the report if you have a look. If not, I will check. But in our new reporting, we should just make a note of some of the common contaminants. We will also remove cow and pig, as well as dog and cat, as they are sort of common contaminants that most people are not interested in, unless there is a strong interest to know, if there is those species. But it's quite uncommon to get that request.
That's brilliant. Thank you, Benji. That was just a general public question about, well, why haven't you found this?
Yeah, of course.
No, it's great. That's absolutely fantastic. I mean, there was some consternation about the relative lack of bird species, and I did explain to people that the birds actually had to be in or using the watercourse in order to detect.
Correct.
Them. So just because the higher diversity of avian species is much higher in the corridor, it doesn't mean that their DNA actually gets into the brook. But one of the questions that came up was how many days or weeks or months roughly before the sampling date, which in this case was at the end of March this year, are we sampling over? So are we, and I know that's a very open-ended question, but thinking about perhaps the vertebrate DNA, are we looking at what was in the watercourse over perhaps the last on that day, or perhaps over the last week, or does it extend to several weeks or even several months? That was a question that came up a couple of times.
Yeah, no, definitely. I'll answer that one, and then I'll just do a brief mention on birds as well, since it came up. So regarding what we call the sort of DNA life, sort of in the water, the advantage of working with water is that it is a DNA degrader. So basically, DNA degrades pretty quickly in water courses. Most of the research data points at sort of between 24 hours to about a week. And most of this depends on the environmental variables. So for example, as you may know, heat and UV will degrade DNA faster. So winter, we might see DNA last a little longer. In alpine lakes, it's been proven to remain in the water up to a week or more. But in most rivers, sort of UK examples and research shows that it does degrade fairly quickly from the point of entry or the point of detection of the fish or the mammal or the bird downstream. So as the DNA moves downstream, it breaks down, and slowly that DNA signal decreases in what we sequence. So the presence of a species in, say, site D will probably likely have occurred just upstream of that site. The DNA will have moved down after the animal in some way came into contact. Rain can also act as a sort of as a help to gather DNA from the surrounding environment and sort of move it into the river course where we can pick it up. And it will quite quickly degrade. So there's lots of experimental data where they have, for example, set up cages with a known species that is absent from that river course, and then they take DNA samples every a couple hundred meters. So that there's lots of nice publications showing that basically within 500 meters, to a kilometer in most rivers, that DNA will be nearly completely absent from the environment.
That's really useful and fascinating, Benji, because people were saying, well, why have you bothered taking 6 samples? Surely the samples at D, E, and F will have integrated the DNA, from everything upstream at A, B, and C. And I said, well, I don't think so. I think that, it degrades sufficiently rapidly that we're really taking a snapshot over a relatively small time frame and relatively local to the sampling site. So
yeah, exactly. And
really you were going to give me a revelation about birds as well.
Well, a revelation is a big word, but yeah, I mean, basically, birds tend to be slightly underrepresented in general from our data. So from the vertebrates, together with reptiles, they're also a group that does not secrete much DNA just because of their lifestyle and their physical a way of life. So basically we see less DNA. So with birds, you'll notice that there's quite a few of the birds that will come into contact with waters, such as rails, herons, geese, duck, and then you should also get some of the maybe some birds using those river gallery sites, but it'll be nearly impossible to get the whole sort of representation of sort of bird species at the site, and it's not really comparable to sort of human effort because, as we all know, where there's lots of great and simple ways to gather bird data at a sort of low cost using experts. So we don't usually recommend eDNA as a way to try and replace that, but rather it can complement. As you've seen here, you picked up a new species. And it can sort of nicely complement some of the sort of species that could be missed and are sort of river or sort of freshwater specialists. So that's just a little side note about birds, and we see the same in tropical environments and sort of lots of diverse environment, wetlands, where we work with this type of data.
It's absolutely fascinating. One of the questions that came up at the BioBlitz event as well was, would the time of year that you did this survey make a difference? And would, somebody said to me, surely the end of March is an awful time to do a survey like this. Surely you should have done it in, June or July. And the advice I previously received was that no, actually, winter, spring was actually quite a good time. I don't know if you can say a few words about that.
Yeah, correct. I mean, we like the spring because there's still good water movement. There's some rains, sort of fresh water moving in to help with those sort of additional detections for mammals and other groups. And it also means that sometimes in the summer the water courses can dry up a little bit. The water could become sort of slightly more turbid in some cases. It depends on the environments, I guess. I mean, chalk streams might get quite clean as well in the summer, and from my experience in Cambridge and surroundings, but you will get less of those additional detections if there's low rain, especially as we know in sort of the East Anglia region, the rains can be quite quite low in the summertime. Hence, I think the suggestion, the only reason to take multiple seasonal samples is if you have a very strong interest in migratory species. So if we're working with people looking at trout or salmon, sometimes they will take samples sort of maybe at different seasons of the year to be able to explore things like migration. I saw that there was mention here of eels missing, so it could be also because I'm not exactly sure when the time of year where they might be moving in these water courses. And the other reason is if you're after particular species, for example, crayfish, we recommend surveys in the early to mid-summer, just because they tend to be more active. And if we want to pick up, white-clawed crayfish or people looking at invasive species, signal crayfish, the work in the summer is best in terms of the activity patterns of the species we are after. So we do recommend timing also based on that usually.
That's fantastic. And since you mentioned crayfish, let's move on to the invertebrates because and we were warned that the invertebrate signal might not work quite as we planned. And as you can see from the on-screen map, only 3 of the 6 samples produced usable eDNA, and certainly on the poster, I did put in the poster that there was, there were problems with the with using the eDNA, and that the PCR didn't quite work as planned. And I wondered if you could say a few words about why those 3 samples failed completely, and what was it, something that we did in the sampling, or was it some other confounding environmental factor?
Yeah, so there's a few hypotheses, and we have been sort of refining our process with sort of internal test data to improve the invertebrate pipeline, hence the warnings that probably Katie gave in terms of there is a sort of failure rate that is usually between 20 to 50%. In your case, it was 50%. And this is, we haven't pinpointed the exact reason for this. It's quite a delicate pipeline that we use to be able, to look for macroinvertebrates from water. They're very much, true traces of DNA in the environment. So at the moment, it could be due to a mix of factors that could be both environmental and sort of the sort of sample collection. But I would say that if all the other ones worked and these were from the same sites and the vertebrate analysis seemed to work very well, so I would say in this case it would definitely not be the handling, but it could be some variables that we just can't sort of currently pinpoint. But there is the opportunity to explore this further with the sort of refined pipeline. Hopefully, like if you do continue to choose to work with us on some of the future bioblitzes and some of the other sites, we could still sort of try and advance the methodology for the sort of macroinvertebrates. But I'm curious, to hear more about the results and sort of your views on these.
So the main thing for us, Benji, was that what we didn't have a firm appreciation of was the sheer biodiversity in the insects. And in particular, we hadn't appreciated just how many Diptera and true flies that we had. And what that, as far as we're concerned, we thought we had a reasonable handle on the biodiversity of the insect groups, and it turned out that we recorded less than 10%. Using direct observation compared with the what was shown by the eDNA survey for the from just 3 sites rather than 6. And so it makes me wonder, wow, if all 6 had worked, what might we have found? So I wondered if that kind of revelation about the the sheer biodiversity, probably of small or hidden, cryptic, difficult to see species. I wonder if that was a common thing that happens in these types of surveys.
Yes, I mean you're definitely correct. It's something that we see reoccurring. That's why we're desperately trying to improve the pipelines so we can get a higher success rate, because this data is proving to be quite it seems to be revolutionizing a little bit sort of how we can approach sort of freshwater invertebrate monitoring in general, because a lot of the scoring methods sort of in the UK have been based on the groups that we can sort of more easily identify, by human eye, by sort of MISNet, by sort of methods like kick sampling and sort of sorting through specimen. But this has probably, neglected a lot of groups that are sort of could play an important role in in these freshwater systems. So definitely, the Diptera are really interesting. Unfortunately, we tend to sort of get a little less of a grasp on some of the sort of harder bodied sort of insects. So this tool is still very much complementary to human effort, we believe. But it does add that sort of incredible additional layer of data. And it seems from your case that you actually got a good diversity of beetles and hard-shelled species as well. So that's really interesting to see. And this poster is very interesting. And if you're happy, I can also share it with our folk internally, just to showcase some of the results from your project.
Yes, for sure, Benji. I mean, obviously I created it it's very simplistic, but what it tries to do is boil down the the information in a way that the public can easily digest it. What I'm going to do is let's just stop that screen share for a second, and then I just wanted to try and share. Let's try this one. Okay, so what you're looking at here now is the output from the most recent BioBlitz at Darien Meadow, which is an urban meadow in the just to the south of the centre of Cambridge. And this is where a team of experts over 24 hours try to record as many different plants and animals as possible. And you can see on the screen in front of you all the birds. And if you scroll, here are the mammals that we did with small mammal trapping. And we only got 3 species of fish. But some of the things I wanted to highlight to you, and please don't think that this is a complaint, but I find it really, really fascinating that some of the commonest invertebrates, and I think the ones you're calling the hard shell invertebrates, perhaps, are not coming up in the eDNA survey when we know they're there. And for example, the water mites, the arachnids, the hydrocarina, certainly we've got diving beetles, certainly we got cased caddis. I'm amazed that we don't get freshwater shrimps, these crustaceans, or indeed the water hog louse. We certainly did get copepoda, the cyclops, the water fleas. There are definitely damselflies and dragonflies using the brook, but they didn't come up in the survey. There are things like greater and lesser water boatmen, the true bugs, clearly leeches. There's a much greater diversity of mollusks, including small clams, these peacockles, and a wide range of snails. We didn't really get a sense of that, and so on. But certainly, you know, our attempts at recording the diptera, the true flies, are quite poor compared to what with the EDA. And of course, you know, I can scroll down and you can see there's lots of extra bees and beetles and butterflies. I wouldn't expect these to be
more terrestrial,
in the terrestrial. So I just thought I would ask your reaction, because what was amazing about the invertebrate eDNA was how many species it uncovered for us that we didn't know. It confirmed quite a few. And then there's a whole range of species which are common, ubiquitous, and we're not getting their eDNA, and there must be a reason for it. And I need to be able to tell me about it.
So there's a couple possible reasons, but it is a common theme that we see across projects. And that's why I mentioned earlier, you know, the tool is highly complementary to, you know, human sort of methods that have been used up to now, sort of, you know, that we talked about. And this is because we feel there's a number of reasons. One is the choice of the primer, which is the sort of the sequence that we sort of analyze. In this case, it's a sort of it's a broad invertebrate primer, and it will tend to amplify DNA of, you know, particular groups just because they have a stronger affiliation in terms of the sequence that we use. So just as an example, for mollusks, we have a completely separate analysis that we could run on your samples that would give you a lot more hits, just because it does focus a lot more on the DNA of mollusks, more sort of, it's a more refined analysis that will exclude everything else. And because those you know, diptera and midges tend to overwhelm some of the samples, though, that will cause some of the more sort of rare sequences that are found, you know, possibly many of those species that you mentioned that were absent could have been there, but in the DNA amplification process, if you have a very high density of DNA from some of the diptera, it will tend to overwhelm everything, and that's why we pick up a lot of those. So we still don't have a fail-proof method, and, you know, this is part of a long-term learning process for us and for all our clients, and that's, you know, part of the interesting, you know, component of using these methods. But what could be interesting is testing out, you know, these samples of yours are now stored with us. So if you did want to run, you know, maybe on those 3 samples, run a mollusk or sort of another specific, a crayfish specific, or any sort of I can send you a list of what we currently can do. We're exploring.
So Benji, can I ask, when we started off with this, we were warned that, for example, the generic invertebrate primers might not pick up crayfish.
Yeah.
And, I'm guessing here, I'm taking a punt, that the crayfish primers might also be useful for other crustaceans. Is that right?
To some extent, yeah. I think the crayfish ones are single species specific. So we tend to amplify only white clawed or signal, but we do have a unionid, you know, primer sort of a Dracaenidae primer. So we have primers that will focus on some of those groups. Usually these are developed based on, the wider interest in our client base. So obviously the invasive species ones tend to dominate just because there's an interest from water companies and consultancies to pick up the sort of invasive non-native species. But it's something that we have been exploring, and I was just mentioning earlier, we are looking to also offer a broad eukaryote freshwater primer, which is going to be quite interesting. We haven't tested it out much. We've used it in marine environments, and it's really interesting what we can get in terms of broad data. And the other thing, a lot of people are using this data also to define, to baseline and look at change in their systems. So because there's so much data, we can use it, like if you were to say do this every year, then you could look at any sort of changes in some of the key groups that you pick up and explore, if those are linked to any impact from other sort of, anthropogenic pressures, fertilizers or whatever is being used in your systems that you're trying to fight for restoration. So it's a way of using this sort of big data approach, just because we get so much, and maybe some of those midges and some of those diptera can actually give us a strong signal of what is happening. We just still have to learn. But as I said, I think it's complementary still for the time being, especially for some of those groups that historically have been used to define things like freshwater health. Those will still need to be, searched for with some of the manual methods, which is great. I think it's, that's where the strength lies here. We always work closely with freshwater sort of ecologists, and that sort of is the power. It's an additional tool in their toolbox to really sort of go out and get the best data that they can from these sites and try and put it together as you have done. It'd be interesting to see once you add the sort of eDNA data into that spreadsheet of yours and see sort of how it can complement your sort of wider.
I tried it, Benji, and you can imagine we've run 6 similar BioBlitz events over the last few years, all the way along the length of Hobson's Brook, and every time we do it, we get about 200 species. But the evidence out there from people who monitor plants or birds or what have you is that actually the BioBlitz data is a small fragment of the total biodiversity, and the eDNA data simply underline the fact that whilst a BioBlitz event is fantastic for public engagement and making a visible point about biodiversity in the landscape, the true biodiversity is much higher. I have a question, which, and this is probably, we'll probably tie it up after this one, I think, because you've been very
I do have to run because I have a plane to catch soon.
I don't know if you can see on the shared screen the site labeled F here is a more it's more like a series of linear ponds rather than a flowing stream. This area has been highlighted previously as a hotspot for dragonfly and damselfly biodiversity. And one of the things that, as I say, I noticed was that we didn't get any, I don't think, of those taxa coming up in the eDNA. And I wonder, presumably it's because the nymphs are hard-bodied, but I wonder if there was a specific primer or something that could be applied to this area. And somebody also asked me, just because the sample from down here at F and at C and A failed for invertebrates, does it mean that the sample was completely contaminated, or could it be that in the fullness of time those samples could be re-examined and the problems ironed out? So sort of 2 questions to end on there.
I think the first one regarding damselflies and dragonflies, it's reoccurring in most projects. It's very hard to pick them up.
Okay.
And I think it is just because of their, their physiology, and they probably just don't they don't secrete much DNA. We're not entirely sure. I don't know of any research, but it's an interesting question. You can probably, have a look online. I'll do so myself. If I see anything interesting, I can send it over. But we have had some requests, because obviously they're also important in tropical systems, to assess. So yeah, hopefully something will come out in the future. And this answers the second question, by which, yes, the DNA is stored. There's a limited number of analyses that can be done. We're usually looking at 3 or 4, just because the DNA extractions then will run out eventually, because we use a little bit for every analysis. But this means that if there are sort of advancements and we feel that we could repeat this work, then we could possibly look into rerunning those 3 sites when there's maybe during the next analysis, and see if with the new procedure we're able to get it. But again, there will be some caveats there, just because it's something that we are still working on. But the DNA is there. We can also, if as I said, if you were interested to run a muscle specific analysis or something like that, I can send you the list of what we currently have in our pipeline, and we can go from there.
And I guess that would be helpful. And I guess that as time goes on, more primers are added to the list.
Exactly,
and,
more primers.
What we tried to do here was just a generic. Nobody had done this before. We think it's the first time that the whole length of the chalk stream has been done. We'd really love to be able to get some of the sites which didn't work this time for invertebrates to work, and in particular the source down here, site A, the source of Hobson's Brook is a beautiful site called 9 Wells, and it's where chalk water from the limestone bubbles up underground. And one of the persistent questions here is there is supposed to be a very particular fauna associated with the spring head, the chalk spring head, because the water comes out at a fixed temperature of about 10.2 degrees C, and it's
we should try
a particular chemistry, and in particular there are some flatworms, some planarians, and also some very particular caddis. There's a species called Agapetus, which are associated with these chalk spring heads. And of course, we'd hoped that we might be able to see the eDNA for these invertebrates in that sample, and of course we didn't. So there's every chance that we may well be able to repeat this survey.
I'll keep you updated, and you can do the same if there's any further questions by email. I do have to run because I'm catching a flight to London from Copenhagen in a couple of hours, so I need to get on the move. But it was great to have this chat. Great to see this poster. If you're happy with me sharing it, I'll share it.
Absolutely happy. Thank you, Benji.
For now, and then if we do want to develop a case study or something, that's something that we can explore as well. If you're interested and do a joint case study, it's something that you might have seen on our website. You can have a look at some of the others we've done. There's actually
yes, they're great. Absolutely.
So we can explore something like that, and let's be in touch about sort of how how you'd like to proceed with this. And Katie will be back hopefully in a couple of weeks, so she will still be the lead, and I'm happy to support.
Well, look, Benji, thank you so much for taking the time to talk me through those things. And if you're happy, I will share this short video to the trustee.
Yeah, of course. And sorry for the
Conduit Trust.
Sorry for the technical glitch.
No worries.
Okay.
Thank you so much for your time. Please keep in touch.
Yeah. Ciao.
Thank you. Take care. Bye-bye.